Welcome, everybody. It's my pleasure to introduce Samir Chowdhury, and today he'll be telling us about exploring the landscape of brain dynamics during ongoing cognition using Mapper.
Thank you very much, Henry, and thank you very much to the organizers for giving me the chance to speak here today. So this is actually a very empirical talk going into some of the successes that Mapper has had in precision psychiatry. And this is all joint work with Caleb Jeunesse, who is a grad student here at Stanford in biophysics, and my postdoc mentor, Manish Sagar. So let's see. So the principle that we'll be dealing with today is fMRI. And in fMRI, what you do is you start off with the group of participants in a very carefully controlled experiment. And the participants stay inside an MRI scanner while they're performing some tasks. And the scanner is continually taking pictures of their brain. And that's how we have access to some idea of the neural activity that is going on while they're processing these tasks on their plastic iPads. The way fMRI works is, well, it doesn't actually measure anything absolute. What it measures is a relative change. So the idea is that when there's a particular region of the brain that is undergoing some neural activity, it has increased metabolic demand, and the body compensates by sending more oxygenated blood into that part of the brain. Now, oxygenated blood has different magnetic properties from deoxygenated blood. And what MRI does is it captures this signal loss due to the change in magnetic properties. Because it's not actually capturing, so MRI does not actually capture like brain activity, it captures something called a hemodynamic response, hemo meaning blood. So the hemodynamic response looks something like this, and this entire cycle of blood going into part of the brain and then sort of like dissipating, that takes about 15 to 20 seconds. So whatever data we get from the fMRI, it's brain activity convolved with a low pass filter. Now the fMRI, another part of the fMRI experiment is the experiment design. So typically what we see, or at least what we'll see in this talk, is a block design where the participant is shown a particular stimulus for a few seconds and then a different stimulus for a few seconds, and so on and so forth in different blocks. And this data, after pre-processing, is passed into a general linear model where the ultimate goal is to try and find these neural correlates, parts of the brain, which activate and deactivate in correspondence with these blocks. So that's how fMRI works, and let's see where fMRI sits in precision psychiatry. Currently, this is sort of where we are. We're in the corner of this cube where we have to average across space and time and participants in order to deal with the low signal to noise ratio of fMRI. It's very noisy data, so to average out noise, you typically do a lot of averaging. But this is a problem with psychiatry because any clinician is always dealing with a single patient at a given time, not an average patient. And so there's a large push toward getting into n equals to 1 neuroimaging. So the state-of-the-art mathematical tools are actually not very state-of-the-art at all. They're still in the realm of standard dimension reduction techniques, including the 100-year-old PCA, which is still every neuroimaging researcher's favorite tool because it just works really well. And it has, it's just very easy and transparent to explain to people. The main thing that clinicians need for any kind of mathematical tool is an anchor to neurobiology. And Mapper is actually pretty good for this because Mapper essentially gives you direct ties back into which parts of the brain the data came from. And we're going to be seeing more of this in the upcoming slides. So let's see what Mapper does. Here's roughly the pipeline of Mapper. So you start off in Mapper with a large data matrix, which has say n rows and p columns. The objective of Mapper is to take this very large data matrix and give you a compressed representation as something perhaps combinatorial, like a graph. The good idea here is that these graphs can then be fed into machine learning pipelines for further post-processing. So the way Mapper works is it takes this high dimensional data set, it typically projects it down into a lower dimensional space. Inside this lower dimensional space, a binning procedure is carried out. And what this bin does is it just groups together points which are close by geometrically in this low dimensional space. it may be the case that when you're passing from the high dimensional space to the low dimensional space, there's a lot of projection loss, in the sense that points that were far apart in the original space are now crushed together. So to deal with this kind of problem, what happens is you take these bins in the lower dimensional space, pull them back to the original high dimensional space, and if these points are far apart, then you do an extra clustering step to move them apart. This gives you a set of clusters in the original high dimensional space, and then you construct a graph out of these clusters. So the nodes of the graph are indexed by the cluster indices, and there's an edge between two clusters if these clusters share any points. And that gives the mapper graph. For any kind of domain-specific application of mapper, it is very crucial to figure out what is a good filter function to use. And that's going to be the purpose of this talk today, to try and find good filter functions for neuroimaging. We also know that certain mapper principles are really useful for neuroimaging. One is that a mapper graph has very low sensitivity to noise, which is good because the data is very noisy, and it produces a compressed representation, which is good for clinicians who want to really understand what they're looking at. Okay. Today we'll be working with a very particular type of experiment design. This is a continuous multitask paradigm. And here the idea is that the participants are going to go through these 4 mental states in succession. So in this data set, there are 18 participants who were scanned continuously for 25 minutes and 24 seconds. And they went through periods of rest and then a working memory task, which is kind of interesting. It's a 2-back working memory task where you're given a sequence of images. And at any given point of the sequence, you're asked if this is the same image that you saw 2 images prior. So you have to sort of like keep it in your working memory for a little bit. And then there's some simple arithmetic tasks and some visual attention where you just look out for a crosshair appearing on a clownfish or something. And each of these tasks is 3 minutes long, and they appear in 2 blocks. This data is ultimately, after a lot of pre-processing, which really takes up the majority of the time in the fMRI pipeline, is transformed into this setup where you have 1,000 rows or observations. And the data is put into 3 instant 5-dimensional feature space, which is what happens here is you take the individual voxels that come from the fMRI data and you push them into biologically predefined regions of interest. So you have, this is the ultimate data before it goes into the mapper pipeline. All right, so we're interested in figuring out good filters for mapper. So let's see what has been done in the past. It turns out that mappers enjoyed quite a bit of success in neuroimaging. So in these studies, what happened, these are also studies that came out of Stanford, and what was going on is these researchers, they were looking at groups of very young boys between 2 to 4 years of age, roughly, with Fragile X syndrome. This is one of the very few known genetic markers of autism disorder. And in a longitudinal study, it was found that mapper revealed these 2 groups. And these groups actually were shown to have different clinical phenotypes. One of them turned out to have high functioning autism. And they also had very neuroanatomically distinct characteristics as well. So specifically, mapper found these groups at the beginning of the longitudinal study, and these were tracked through time. So mapper gave a good way of looking at these subjects. Here, the experiment was at the group level. Each row of the data matrix was a particular subject. At the n=1 level, more recently, mapper was applied to the continuous multitask paradigm from the previous slide. And here, there were no patient populations. These were people, I think, between 25 and 30 years of age. And it was found that the community structure of the mapper graph that was produced actually was tied to their performance, the performance of these participants on the different tasks. So in particular, mapper graphs with higher community structure, as in this case, were correlated with higher performance in the tasks. And this is a mapper graph coming from a low performing subject. And you can see that the different tasks are sort of like squished together into a single blob. whereas over here, the different tasks are actually sort of separated out in a graph. The idea here is that participants who were able to switch on and off between different brain states that are particularly useful for certain tasks were able to perform better, and this is reflected in the mapper graph. The specific filter here that was used was one of the ISD neighborhood lenses, which is a variant of stochastic neighbor embedding, which is a nonlinear filter. And this gives us an idea that perhaps we should look into these linear versus nonlinear dimensional reduction techniques. So the simplest access to getting a nonlinear distance is the k-nearest neighbor graph. The way this construction proceeds is as follows. You start with an n by n pairwise distance matrix using the ground metric that you're given. You fix some integer, and then you build a graph on your n nodes with an edge whenever between any two nodes whenever these nodes are the top k nearest neighbors of each other. And this gives you access to a new distance matrix comprising the geodesic distances between the points on the graph. And if you perform one more post-processing step by taking an MDS projection of this new geodesic distance matrix, you get what is known as Isomap. And the classical example to illustrate the usefulness of Isomap is in showing that it can unroll this Swiss roll dataset nicely into the plane. There's some potential issues. There's some topological instability that comes with Isomap, but actually Mapper, with its step of doing partial clustering in the high dimensional space, can try and resolve some of these topological instabilities. All right, so we're trying to ascertain how well these linear versus nonlinear filters work, how discriminative these distances are. So we actually use a technique of distance distributions, which have been shown. It's been shown that distance distributions, basically the histogram of distances that appear in your data set, can characterize shapes up to rigid transformations with a few exceptions. So what we did was we did the following. We first looked at the distribution of distances between tasks using the original ground LP metric, in this case the L1 metric, versus the distribution of distances between tasks using the geodesic metric. And these distributions are actually quite a bit different, quite more different for the geodesic distances, showing that there's these geodesic distances that are more discriminative. So that was a good indicator for us that we should be using some sort of nonlinear filter. The next sort of step that happens in the Mapper pipeline is to figure out what parameters to use. So Mapper comes with parameters which control the granularity of the Mapper graph. The interesting thing with neuroimaging data is that this hemodynamic response, because fMRI measures actually changes in blood concentration, this hemodynamic response takes about 15 seconds to wipe out completely and acts as a physical low-pass filter. And therefore, it imposes autocorrelation structure on the data. And if you're not careful, if your binning is somehow too local with the Mapper graph, you get a graph that looks sort of like this. And all you're really picking out is the temporal structure of the experiment design, which is not very interesting to have. So to deal with this, we did something very simple. We create this sort of incremental heuristic algorithm, which what it does is it estimates the autocorrelation structure of the data, or this can also be fed into the pipeline directly because you have knowledge of how much time elapsed between each slice acquisition in the fMRI. In this case, it was 1.5 seconds. And you know that the hemodynamic response peaks at around 11 seconds. So the idea is you fix some parameter tau, and then you say that you incrementally make your Mapper graph more and more global until at least alpha percentage, alpha in this case was set to 30, until 30% of your final Mapper graph nodes contain 2 points that are at least tau acquisitions apart. The idea is that this acts as some sort of critical scale which makes sure that the graph is not completely driven by the autocorrelation structure of the hemodynamic response. And these two steps, the nonlinear filter and this sort of autocorrelation dependent parameter choice, it works well because it actually validates the results that were obtained previously using the ISD platform. And here we get these Mapper graphs, and here these visualizations are performed using Dinosaur, which is a neuroimaging specific extension to Kepler Mapper. And what we see once again is we recover the characteristics that high community structure is correlated with high performance in the different tasks. And in addition to this, we were also looking at different post-hoc analysis methods. Now, the nice thing with mapper graphs is that they can be fed into machine learning pipelines, or they can just be analyzed using many of the different network science tools that are available. We also saw that something that has been used in the past for studying mapper in the neuroimaging context was to take a particular region of interest and look at the distribution of distances to other regions. And in this paper by Alice Pitania and collaborators, they have these nice ridge plots where they were able to get some insights into the dopamine pathways of a mapper graph. So the formal framework for studying distributions in a mapper graph really sort of like calls for, in some sense, perhaps optimal transport. The idea is that a mapper graph is a metric space with a geodesic distance, so we can use the Kantorovich-Rubinstein and Wasserstein metric to compare the distributions of the different tasks. And so what this metric is as follows. Suppose you have different tasks, which have, suppose you have a bunch of points in some metric space, and the points have distributions on them. In this case, the distributions are shown using the size of the points. And the task is to transport the points, these red points, to the blue points, but while taking into account the distributions. And this can be set up as a linear program, and this metric up here is simply arising as the solution to this linear program. Essentially, what this does is it measures the optimal way to transport one distribution into another distribution while taking into account the metric that you have to use the ground level. And so let's see how this works. It turns out that this gives us more granular information than what was known in the previous state of the art. So you take some mapper graphs and you can look at the different distributions of the tasks. And it turns out that the optimal transport distances between the math and working memory distributions is actually a very good predictor of performance. It finds group differences in the sense that these high-performing groups have much more distance between the math and working memory distributions than the low-performing groups. You can see it over here. This is a high-performing a mapper graph, and this is the working memory distribution, whereas this is math, and here it's much more concentrated, here it's much more diffuse. This is a low performer. Here the math and working memory is sort of like equally diffuse. All right, so in particular, it gives us higher information than just the community structure because it tells us which tasks were driving the community structure. Now to summarize, here's what we did essentially. So we've said that, okay, due to these empirical results, for n equals to 1 neuroimaging, mapper filters, whatever you make, they should perhaps be based on nonlinear distances, or they should at least take advantage of these nonlinear distances. For fMRI data at least, the autocorrelation structure coming from the hemodynamic response function reveals a critical scale for choosing mapper parameters. And these are the things that are sort of critical for coming up with a good mapper graph in the first place. And then for post-hoc analysis, we see that using optimal transport reveals these group differences with higher granularity than was known with the standard community detection tools. Now, we presented some of these results to researchers and clinicians, and here are some interesting questions that we got that are actually can be phrased mathematically. So one question is, how many minutes of data is enough for mapper? And this is important because for certain populations, it's hard for them to like stay steady inside the scanner. So you have very short scans. Perhaps the 25 minutes that we had in our data set can only be something like 6 minutes for a child or a child with autism spectrum disorder. So the mathematical question is, how does mapper work in this vastly undersampled regime? Like, is there any kind of inference that can be made? Another item is that the filters that we used were purely taking advantage of geometric similarity, but now in computer vision, there's a lot of interest in building up methods that take semantically similar points into consideration. In this case, semantically similar would mean perhaps time points, which correspond to similar neural activity. And we would like some filters that take advantage of these advances in the computer vision literature. Another sort of like basic question is, how sensitive is Mapper to standard time series preprocessing steps? So there's some preprocessing that's very particular to fMRI, such as spatial smoothing and so on, but some which are very standard for any kind of time series processing. Temporal bandpass filtering is one, pre-whitening to remove autocorrelation structure is another. And the question here is, the statistical transforms are applied to the columns of data. How does transforming the columns translate to the spatial geometric organization of the rows in the high dimensional data space? So these are some like questions that we're actively thinking about, and any sort of like insight from the math community would be fantastic in these respects. So with that, I think I took up exactly 20 minutes, and I'd like to thank you for your time.
Thank you very much, Samir, for the wonderful talk. Before questions, let's briefly unmute ourselves and applaud for Samir.
Very nice, thank you.
Questions for the speaker? Let me start with one that Henry has chatted in. So he writes, his question is related to your last point about smoothing. When using Mapper to handle fMRI data, is it possible to do subsampling of the raw data, especially when n is bigger than 1? It seems that there are some kind of periodicity, and a subsampling may reveal stronger patterns.
Like that could be very cool, right? Like in principle, I think sure. But I don't necessarily, okay, so for the n greater than 1 level, here's what people typically do. People typically, in many cases, will just concatenate data. This somehow makes some sense if you're using PCA, perhaps. And yeah, you can also like concatenate data in Mapper as well. And perhaps, yeah, this is a great idea, actually, that if you sort of like combine data sets coming in and then subsample to reveal this periodicity, that would be great. Now, here's the fun part though. In this particular experiment, we're using the ground truth, but the real goal is the phone. The real goal is to have participants who are not doing anything at all. They're just resting inside the scanner, staring at a cross, and you're not giving them any instructions. The reason is that the moment you give a participant instructions, they are perhaps thinking of different things. And if their mind starts to wander, you have no control really over what they're really thinking versus what the scanner is picking up and versus what your experiment design is supposed to do. So if these things, if the periodicity and subsampling can be done in a fully unsupervised way, that would be sort of like a gold standard for neuroimaging and for clinical purposes, especially.
Samir, let me give you one more question from the chat window, and then we'll move to some questions from speakers audience members who want to ask out loud. David Snyder writes, was MATLAB used to plot the distance distributions? And as a follow-up question, will the slides or the references be posted somewhere? I don't know if they're up on your webpage, for example, Samir.
Okay, so these distributions are a rich plot using I think, Seaborn. I know that was on the question, but I just want to like give some insights as to like how these plots are done. These violin plots are some MATLAB package I think you can find on MathWorks webpage. These distributions are simply the raw MATLAB distributions, but these are the violin plots again, coming from, I forget which package. But yeah, you can look these up easily. Slides are references. So we're actually working in a bioRxiv publication. Actually, Caleb is working very hard on coming up with validation on separate data sets, because that's like the gold standard to have validation across different data sets. And we hope to release something in the bioRxiv very soon. But yeah, currently these are not public on my webpage or anything. But I guess they'll be public on YouTube.
That's right. So we still have another 4 minutes at least for questions, just because I started Samir a bit later than normal. I see Leonid has a question in the chat window, but before I get to that, any questions from audience members that would like to ask them out loud?
I have a quick question, actually. I'm not sure how relevant it is, but do you think it would be useful or would add anything to try incorporating a delay embedding into the pipeline? Do you think there's any more structure that's there, or at that point it just becomes a mess?
I think it's, of course, like a very natural thing to try, right? To look at delay embeddings and then see what you get. Here's a problem with, and this sort of relates a little bit to what Henry asked, like the problem is periodicity, because usually like finding like periodic data is fantastic, but in fMRI, you have to be careful with the experiment design. So in this particular case, the experiment design was set up so that it was actually randomized. So it's not like you have rest, memory, video, math, rest, memory, video, math. Instead, it's a little bit different. The reason is that if the participant anticipates the periodicity of the experiment, the anticipation will show up as brain activity. And this would be a confounding factor. So you have to be careful not to set up your experiment to be periodic. Having said that, if you were purely just taking resting state data and seeing if there are periodic patterns, that could be useful, because it could perhaps show that participants who may be suffering from depression or something, or some sort of anxiety disorder, could have these periodic patterns of thought that just stay with them. And this is a hypothesis that a lot of people are trying to test, actually. So yes, delay embeddings in general, with a little bit of care as to the experiment design, can certainly be useful.
Thank you, interesting.
More questions from the audience out loud? Samir and Leonid, I think Leonid's question is on a different project. It's on applying Dowker complexes for voxel analysis. So maybe I'll let the two of you follow up.
Sure, I'll take
outside.
that.
Does that sound good? Yeah. Maybe just copy the chat.
Perfect.
And one last question from Ketchi. So did you try this on simulated data?
Quite specifically, not. Okay, there's reasons for and against. So simulated data would be nice because you have full control, and there you can actually like test certain like hypotheses, like how much data is required to get insights from mapper. So that kind of question can be carried out in simulated data. Of course, the gold standard is to use real experimental data, and we were lucky because this experimental data had been collected before. But there's also big challenges there because experimental data is extremely noisy. You have like a 2 millimeter window of motion in which humans will move into the scanner, and that will cause like artifacts and confounds, and these will be picked up. You have to be careful to even exclude cardiac and respiratory confounds from the data. So real data is beautiful because it actually gets us close to what clinicians are interested in, but it comes with challenges. So no, we haven't tried this in simulated data, but definitely it can be answered with simulated data, which would give us insight into how to design experiments for real world settings.
Well, thank you so much, Samir, for the fantastic talk, and to all of the audience members for the good questions.
Thank you for the questions.